Well, hello, students. It's another day, and I'm not running a temperature, and I don't have a sore throat, so maybe I don't have it. But I've been incarcerated in this house, thinking about you. So I'm going to work on Neuro2. I was very heartened in that in Neuro1, I went for over 50 minutes, and I expected some questions, and I didn't get any. So hopefully y'all understood. I also got 33 views, which made me feel good too. So some of you may have viewed it twice. Now, I want to review a little bit of what we did day before yesterday so you know where we are, and the review is good to jiggle your memory and remind you of what you already know. We defined the nervous system as one of the 11 systems that controls the entire body. It does it very fast through the movement of impulses down sensory neurons and interneurons and motor neurons, moves very quickly in the sodium-potassium pump. We said the secondary or auxiliary system after the nervous system is the endocrine system, which uses substances known as hormones, which are chemical messengers. And it takes hormones a long, long time to work in comparison to the nerve impulse. Now, when we study the nervous system, we have to break it down for study. It makes it much easier. And the nervous system is divided, as you know, into two parts: the central nervous system and the peripheral nervous system. The central nervous system, again, is made up of the brain and spinal cord. The peripheral nervous system is everything outside that brain and cord. Now, the peripheral nervous system is then again divided into the somatic and the autonomic. The somatic nervous system are those neurons that we have control over. We control those nerves. We make a decision with our brain. We send it down the brain stem to the spinal cord and out of the spinal nerves, and sometimes the cranial nerves. And these nerves innervate muscles so that I can sit down, talk to my beautiful students, I can hold my pen and go over what I have already taught and am reviewing. So biological feedback through meditation, but for the most part, we don't have a great deal of control. The autonomics, they control things that we need to have done in the fight or flight response. We need that, or in the relaxed mode. So the somatics are those that we control. The autonomics are those that we don't control, and we divide those into the P.S. I love you, parasympathetic and sympathetic. The parasympathetic puts us in the relaxed mode. The sympathetic puts us in the stressed or fight or flight mode. Okay. Now, the next thing we discussed were two types of nerve cells in the entire nervous system. And we're talking about billions of cells, but there's two major categories. The first is the neuron, N-E-U-R-O-N. The neuron is a cell that carries an impulse, and the impulse occurs because of the inward movement of sodium and the outward movement of potassium through the neuron membrane. That is the pulse. That is the nerve pulse, and they also call it the action potential. We said that there were three types of neurons: sensory neurons, which have a lot of diversity because they're built so that they can receive a stimulus, be it light or pressure or temperature. They're built so that they will work. Structure dictates physiology. Those are the sensory. And when the sensory carry the message to the spinal cord and the brain, then we have special neurons known as interneurons, associative neurons, connective neurons that link sensory to motor. And it's these neurons scattered throughout the spinal cord and through the brain that make sense of what we hear, see, taste, touch, smell, and feel. These are the interneurons. There's, remember, 3 synonyms for those. The motor neurons are those neurons that exit, and remember, they will exit the ventral portion of the spinal cord. They go to the muscles and they go to the glands. So it is the motor neurons that relate to one another, motor to motor to motor to motor, finally to the muscle or gland, to get that gland to secrete or that muscle to contract. And they do so with, remember, the neurotransmitters. Okay. Now, the other type of neuron, and we'll talk about it in some detail, is the neuroglia. Neuroglia means nerve glue, and it goes a long way back. The anatomists, once they discovered through microscopy that these neuroglia occurred, they felt that the neuroglia just glued everything together. So that's the reason they called it nerve glue. They felt it holds things together. Well, that's not completely false, because they do hold things together, but they're much more important than that. They protect, they phagocytize, they support, they feed, and hold the nervous system together. But that's just one of many activities that the neuroglia does. Now, we have 3 types of sensory associative and motor, 3 types of neurons, but we have 6 types of neuroglia, and we're going to learn two types to begin with. Now, the neuroglia are interesting. They're fun to study. They're fun to teach because they have a lot of personality. They do a lot of things, whereas the motor neuron just transmitted that impulse. So we will study those today. We also mention receptor organs such as your eyes, your ears, your tongue, your sense of smell. Your eyes are optics, your ears auditory, your sense of smell olfactory, and the sense of taste gustatory. And there's another sense too that I'll mention to you that's really fascinating. That's the proprioceptive sense of neurons in your muscles that are constantly feeding back to the spinal cord and to the brain of where your body is in space and time. So the neuroglia are really interesting. And if I was ever going to do research in neurology, I'd want to focus on these neuroglia. They're so interesting. And as I said, there's 6 different types. We'll study 2 today. We studied 3 of the neurons the other day. We'll study 2 neuroglia today. Now, as I said, these sensory neurons are in receptor organs. We mentioned those receptor organs that house these specific sensory neurons that pick up sound and light, etc. Those are receptors. They carry the message to the spinal cord through these sensory neurons. In the spinal cord, the sensory is linked to the motor in the spinal cord, and sometimes the sensory goes all the way up the brain through spinal tracts, letting your brain know what just happened externally and even internally. But it's the receptor organs that house them. Now, once the brain makes a decision to do something, then the message comes back from the brain down the spinal cord, out the motor neurons, and sometimes it will do it through the spinal cord before it even relays it to the brain. That's in that is in the arc and the impulse arc. But what happens here is that the motor neurons are going to give messages for muscles to contract or glands to secrete. So muscles and glands are effectors. So you have over 600 muscles in your body, and every single one of them is considered an effector muscle. Okay, we also mentioned the other day afferent and efferent structures. Afferent is a reference and can be a reference to the sensory neurons because they're carrying the impulse afferently to the spinal cord and to the brain, afferently, going towards the spinal cord and brain. And the spinal cord and brain makes a decision, and the motor neurons, they exit the spinal cord and the brain exit starts with an E, and so does efferent. So the efferent impulses are the motor neurons that go to the effectors, the muscles and glands. Afferent towards, efferent away. And even when I mentioned that, and I showed you this the other day, you can even talk about it when you're talking about the anatomy of the neuron. The dendrites, and by the way, I was reading the other night, Neil just named a few. A single cell body with these dendrites coming out from it, it can have 500 branches, 500 dendrites that seek to find other neurons or muscles and glands. Afferent structures or processes, afferent, going towards the cell body. The axon is coming away from the cell body, so it is an efferent process. So you can use that term afferent and efferent pretty broadly. But what I introduced to you the other day was sensory neurons are afferent. They're taking the message to the brain and the cord, and the motor neurons are efferent, coming off that brain and that cord. Then we studied neuronal anatomy, and that was when I used this little diagram showing and discussing the significance of the dendrites, the cell body, the axon. These are collateral branches down here, and at the end of each branch, remember there are podia or feet or in-brush fibers, and they're full of neurotransmitters. When they're stimulated by the electrical impulse, the action potential, then they will secrete the neurotransmitter into the synaptic cleft, also called a synapse, or into a neuromuscular or a myoneural junction if they're stimulating another muscle or stimulating a gland. It's called differently, but there's always a space. Remember, the neuron never touches another neuron. Neither does it touch a muscle or gland. It talks to it with these chemicals secreted by the podia. Okay, that was the classic motor neuron. And I said that represents one cell, one motor neuron cell. Now, these little yellow structures that Neil drew around the axon, which you can see through these structures, that axon is an efferent process that carries the electrical impulse to the collateral branches, but they're surrounded, in this case, by neuroglia. Those are neuroglia, nerve glue, and these specific nerve glue are in the PNS. You find them in the PNS, and they're called Schwann cells. And I guess they named them Schwann cells after that scientist, and don't forget, Schleiden and Schwann, that gave us the cell theory. Okay. So that's basically what we did the other day. And today, I'm moving from the neuron, and I'm going to talk about the neuroglia. As I said before, they're fascinating, they're interesting, and there's 6 different types. We're going to focus on 2. Now, if you look at lecture 2 on page 7 in your class notes, under neuroglia, you can follow along with me. Also, your textbook has some beautiful examples of these neuroglia, and seeing it is 1,000 words when you actually see the structure. Now, when we study them, they're in diversity. There's 6 different types, but 2 we'll talk about today. The second type of nerve cell, and neuroglia is a nerve cell, just like the sensory neurons, the interneurons, and motor neurons are nerve cells. They're nerve cells that are neurons. These are neuroglia, and they do not convey an impulse. They surround, they protect, they insulate, but they do not send an impulse. But when present in the axon of a motor neuron, they do allow that impulse to pass through that neuron much, much faster. And so they're desirable. When they're present, the nervous tissue is white in appearance. When they're absent, the nervous tissue is gray, and we mentioned that the other day. Here it tells you in that first couple of sentences about the fact that they support and hold various nervous tissue together. They carry on phagocytosis. Yes, some of them in the brain can actually move around through the brain just like a white blood cell that engulfs a bacteria in your blood. Some of these neuroglia can move around through your brain and actually phagocytize debris or a pathogen. These neuroglia also supply nutrients to new neurons, which connect them to the blood supply, because all of these nerve cells all need oxygen and glucose and amino acids and minerals and vitamins and fats, just like any other cell in your body does need. As a matter of fact, your nervous tissue in your brain is so needy of oxygen that if the brain cells go for 2 minutes without oxygen, they begin to die, or they can have damage, and sometimes permanent damage. And if they go for 4 minutes, these neurons can have brain death. Those neurons will die. They're so dependent upon that oxygen content coming to them through the oxygen, the hemoglobin in your red blood cells, carried to the brain by two of the carotid on the right and the left. Okay, now, under number 1 and number 2, I mentioned the two neuroglia we're going to mention today: oligodendrocytes and Schwann cells. The oligodendrocyte, I know it sounds like a big word. Y'all are learning so much new words. It's so exciting. And you can impress your family with all these great words. Oligodendrocyte is a type of neuroglia found in the central nervous system, whereas the Schwann, Schwann, is found in the peripheral nervous system. Okay. Both of these cells wrap around nerve cells or neurons. They wrap around the axons in the CNS and PNS, and they have high concentrations of lipoprotein. Now, lipoprotein is a lipid bonded to a protein, and these lipoproteins in the neuroglia, the presence of these lipoproteins in the neuroglia have a term or name for this specific type, and they are myelin, M-Y-E-L-I-N. So the fat in the neuroglia is myelin, and that myelin is made by the Golgi apparatus. Remember the Golgi apparatus that we discussed in cytology and a little bit in histology and throughout the other systems. The Golgi apparatus, they make that myelin, they make that lipoprotein. And of course, the Golgi apparatus, these cells get that nutrient from the blood supply, from the diet. They actually have a fatty appearance, which is what gives the neuron or the nerve its white appearance. All right. Let's start out with the Schwann cells, and then I'll go to the oligodendrocytes. It's reversed in your notes, but I think I'll go to the Schwann first. Now, we looked at Neil's drawing, and this is a motor neuron, and we know it's present in the peripheral nervous system outside the brain and spinal cord, because here are the characteristic Schwann cells wrapped around that axon. You don't find Schwann cells except in the peripheral nervous system. Now, they're really quite interesting. I'm going to show you another picture of them. Just be a little patient. I'll show you another picture of them. I sure miss my overheads. Okay. Here's the other picture of the Schwann cell. Now, what the artist has done here is the cell body. These are the dendrites. Here is the axon, and we've cut off the axon. Okay, we've cut it right here so you can study it up close. This blue structure running through the center, which you can see, that is the axon right there. And wrapped around the axon is the neuroglia. Here, since we cut it off here, we see 2 neuroglia cells. Now, the interesting thing about a neuroglia, it's a cell just like any other cell, and it also has a nucleus. Here you can see the nucleus. You could probably see this in your textbook better, but there's the nucleus that belongs to this neuroglia. Here is the nucleus that belongs to this one. And what happens is, in the formation of these neuroglia, they seem to roll themselves, these layers of fat roll around the axon, kind of like, the hors d'oeuvres, or the pig in a basket, or the, what do they call those things, where you put the little sausage and you roll the dough around it and bake it in the oven. I can't think of the name of them, but you roll the sausage or the dough around the sausage, just like the myelin rolls around the axon. And you can see the layers right here. You can beautifully see them in your textbook. There's the nucleus, and these are the layers rolling around the axon. And that is referred to, this area where you have the concentration of myelin is the myelin sheath, the myelin sheath. Now, what is interesting, the myelin sheath is that lipoprotein. What is interesting about the Schwann cells that you do not see in any of the neuroglia cells in the brain or spinal cord, you only see them in the Schwann cells, is a layer referred to as a neurolema. And it is this outside layer or membrane around the myelin. That is referred to as the neurolema. It's very important, and they've been doing lots of studies with them and with the chemicals that make up the neurolema, because we know that if you injure a nerve or a neuron in the PNS, in the peripheral nervous system, it has an ability to regenerate. It can regenerate that entire nerve. It's fascinating how it does it, and I'll show you a picture in a moment. But it can actually slowly, progressively regenerate. That's the reason if you injure a nerve or neurons in the PNS, you can regenerate them. You can, and slowly, but it does work. And it sometimes, if you make an injury and a cut, that area may seem numb for a while, but after months and a few years, it just slowly regenerates and establishes itself again. And I'm going to show you another picture of that myelin and that neurolema. Where have I been going now? That myelin and the neurolema, because I want you to have a real good visual of that. Here is another example of it. Here's just a piece. There's the axon. Here is the entire Schwann cell, which is wrapping around the axon and making these layers, as you can see. And this outside sheath is what they referred to as the neurolema sheath. Neurolema, neurolema sheath. The fat right here is myelin, but this outer layer here is the neurolema. Very important, because we know it can regenerate. Now, I have a picture that I laid out that I do want to show y'all, which actually shows how these wonderful little neurons and nerves can regenerate. Okay. All right. This shows, I love this. This is the events that occur when a myelinated axon is injured. Now, there's a new word for you. We know that the fat in the neuroglia is known as myelin, the lipoprotein myelin made by the Golgi apparatus. And when a motor neuron has these Schwann cells around it, it is referred to as a myelinated cell. The axons are myelinated and white in color. Now, here is the normal motor neuron, and here is the gap. Notice there's a gap between all of these neuroglia. That gap is referred to as the node of Ranvier. Now, the Europeans pronounce it a little differently, Ranvier, because it's a French word. Of course, I'm sure he named himself after it. But that's the node of Ranvier. And the beauty of it, as I mentioned earlier, is that the electron impulse, or the electrical impulse, is fast in a myelinated motor neuron because it jumps from node to node to node. These are nodes of Ranvier, and the impulse jumps or leaps. That's where they call them saltatory jumps or saltatory leaps. And saltatory means a leaping impulse. When I first heard that, I thought, oh, well, that must be because the sodium ions are on the outside and sodium is in salt. Good try. But it actually comes from the concept that the impulse is jumping from node to node to node. Now, this classic motor neuron is taking the impulse from the dendrite to the cell body, down the axon to the collateral branches, and this represents a muscle. So this is skeletal muscle, striated muscle. You can tell it's striated because you can see the myelin and the myosin and actin concentrations. And here's the collateral branch, which will release a neurotransmitter to this muscle, causing it to contract. Now, that's what one looks like intact. Up here, the artist is showing you that right here is a site of injury. That's where the axon has been torn and disrupted. Now, what happens in this progress, he's showing you how it really regenerates. If you look at the second illustration, here's where the axon was broken, and these cells are beginning to die. These are neuroglia cells that look like they're beginning to die or shrivel up. Now we'll go to C, and you'll see what is happening is that maybe the myelin was disrupted, but the neurolema is intact here and here. And the neurolema, which was responsible, it's the sheath that actually causes the neuronal regeneration. The neurolema begins to proliferate and start to slowly build that portion of the motor neuron. It reestablishes itself here as the Schwann cells, and it reestablishes the axon. Up here you saw that the axon is kaput, it's gone. The collateral branches are gone, but down here, because of that neurolema, it's reestablishing that axon and those collateral branches. One thing I want to mention that just crossed my mind, and I told you that when we first started studying cells, that the jelly that fills up that cell is referred to as cytosol. And when you have the organelles floating around, it's a cytoplasm. When we studied the muscle cells, we call the cytoplasm sarcoplasm. And when we study neurons, they have cytoplasm, but it's called axoplasm. And that axoplasm streams down from that cell body all the way down the axon, that axoplasm. And you have all the organelles here in the axoplasm, the endoplasmic reticulum, the Golgi apparatus, lots of that because you're making lots of lipoprotein. You have the lysosomes, you have the nucleolus within the nucleus, everything's there. And this is a complete cell, except that you have one motor neuron and neuroglia, which are slowly reestablishing themselves. This study, and I'll mention this because it's kind of a fun fact, this study of neurolema has been going on for some time, and we do know that some animals, some invertebrates, are actually able to regenerate. Starfish, for example, a starfish that has various arms emanating from a central disc. If you chop off the arms of the starfish, but you leave part of that central disc present, then over time the starfish will regenerate new stars, new legs that come out from that starfish. They'll regenerate. And a few decades ago, they found out that the Great Barrier Reef off the coast of Australia was losing a lot of the coral reef because there was a starfish known as the crown of thorns starfish that was actually climbing all over that coral reef, sucking out the polyps that made the coral reef alive and killing the reef. So they decided that they had to get rid of the crown of thorns starfish so that they could survive these wonderful coral reefs that go out for miles, and they're magnificently beautiful. So they had high school students go out in trawlers, and the boys would, and some of the girls too, they would dive down, and they would they had gloves, the water was pretty shallow, they would dive down, pull up the starfish and put them in bags, and then send them back up, bring them back up, and they would put them on the deck. And of course, all the kids had boots on to protect their feet and legs, and they just chopped up all the starfish. And then thought, well, we killed them, threw them back in the water, back in the ocean. What they didn't realize was they were just propagating them because they chopped them all up and they just made maybe 10 starfish from the one. When they realized that, they had to go back and adjust. And then the students would dive down into the water and actually inject a toxin or an alcohol, rubbing alcohol, into the starfish and kill it. And that was the only way they could kill it. They couldn't chop it up. That was a big mistake. But they do know that there's some chemical in the starfish, maybe very similar to the chemicals in the neurolema, that can actually cause that regeneration. Okay, those are Schwann cells. Now, I want to mention a little bit more. I want you to really understand the nerve impulse. So I'm going to talk about it just a little bit more. And I want you to see it. Okay. Now, right now, don't worry about the impulse. What I want you to focus on is this is an axon. Here is 1, 2, 3, 4 neuroglia. And I want you just to look at this structure right here. Of course, there's the space between the neuroglia, and this is called the nodes of Ranvier. And all along the axon, you have lots and lots and lots of sodium atoms. And the sodium atoms are charged electrolytes. They have one more proton in the nucleus than electron. So all of the sodium outside of a motor neuron, that sodium has a positive charge, and there's a lot of it. Now, in addition to that positively charged sodium, there's also other negatively charged ions. And the negatively charged ions have one or two more electrons in the energy shells than they have protons, so they have a negative charge. But they're on the outside and they're negatively charged. Well, if there was the same amount of negatively charged ions as there were sodium ions, which were positive, they would negate one another and there wouldn't be a charge. But there's more positive than negative. So the positive charge prevails. And in a resting neuron, the sodium is like an alley cat, cats that love to live outside. They like to stay, for the most part, on the outside of a cell. And there's lots of them. So there's a lot of positively charged sodium here. On the inside is potassium. Now, potassium, it shows negative here. Potassium's not negative. Potassium is also positively charged, but there's fewer positively charged potassium ions inside the axon and more negatively charged ions. Sodium phosphate is an example. So there's more negative than positive potassium inside, so it's negatively charged when it's in the resting state or resting potential. The outside's positive. Now, when the impulse occurs at the dendrite, the impulse, and I'll mention this to you because I want to make it so clear. I want to make it clear because when I first heard this, I was confused, and it took me a while to figure it out. And I want you to be able to get to see this. On the outside is the sodium, and more sodium, positively charged ions than negatively charged ions. When the impulse begins at the dendrite, the sodium goes in. Now, it takes energy in the form of ATP. It takes active energy, and remember we talked about active and passive energy. Active energy to pump that sodium in, and it pumps the sodium in and the potassium out. Okay. And that's referred to as the classic sodium-potassium pump. These are the two electrolytes involved. Now, sodium and potassium are able to go inside the neuron. Sodium goes in, potassium goes out, and then it can switch again. The negatively charged electrons on the outside, electrons are negatively charged. These sulfates and phosphates that are negatively charged, they can't go in. The only thing that can exchange is sodium in, then potassium out. So sodium has many more numbers. So as the sodium is pumped inward, there's so many sodium that they overwhelm the electrons inside the axon, and the charge inside becomes positive. When the potassium is pumped outside, there are fewer potassium, many fewer potassium pumped outside than sodium in, and the charge of the negatively charged sulfates and phosphates, then they take over. So sodium goes in, it becomes positive on the in, and potassium goes out because there's not enough to overcome the negatively charged phosphates. They take over. So it's sodium in, potassium out, sodium in, potassium out, and the change is made. The exchange is made, and the impulse changes. So that's what they refer to as an electrical impulse or an action potential, or the wave of electrical negativity. That's what they're talking about. Only the sodium can go in and the potassium. And right behind that exchange it recovers. It has to recover in order to get another impulse. It has to recover, or you can't put in another impulse in that motor neuron. And then when the neuron gets all the way in to the collateral branches, they then enter these little podia. The stimulation enters the little podia at the end of the collateral branches that we call podia, synaptic knobs, in-brush fibers. We refer to them little feet, and they're full of neurotransmitters. And I have a picture of that. Let me see. I do want you to see that. Okay. And now this is an actual micrograph of that. This is a great shot. Okay. Here is the little podia or feet, and they contain, they're full of organelles too. These are mitochondria. Each one is a mitochondrion. Remember the singular. These are little vesicles. They call synaptic vesicles, and they're full of what? Neurotransmitters. And remember the most common we studied so far that we talked about, acetylcholine? They're full of them. And when this motor neuron is stimulated in the collateral branches and stimulates this little podia, then the podia that contains the vesicles will actually migrate and commune with the cell membrane, and in a process known as exocytosis, it will secrete the acetylcholine into the neuromuscular junction, if this is a muscle, or the synapse, if this is a dendrite of another motor neuron this motor neuron is talking to. So that's what it looks like. And this is actually a transmission electron micrograph of the synaptic knob filled with these little vesicles. Remember, the most common neurotransmitter throughout your body that stimulates your muscles to contract and your glands to secrete is acetylcholine. Again, remember there is an enzyme neurotransmitter that is secreted by the muscle itself, where this podia is attracted because it has a receptor site, and another neurotransmitter, which is an enzyme because it ends in ASE, enzymes end in ASE, that is acetylcholinesterase, which is secreted into that cleft and stops the impulse. So, but you can have a number of impulses that keep coming and coming if you have a tetanic contracture. So we talked about the isometrics, isotonics, in the three types of contracture, or the tonic contracture. But that's what happens. Remember, the podia do not touch the muscle. They do not touch the dendrite of another motor neuron they're speaking to. The connection is made by the neurotransmitters. And since those words take 1,000 words to describe a picture, here is another example of it. Just look at this part of it. Here's the nerve impulse coming down the axon or the collateral branch, stimulating the podia to secrete the neurotransmitters into the, and this is referred to as a synaptic, postsynaptic membrane here. Here's the synapse. This is between two motor neurons. Here's the synapse or the space. Here is the dendrite where this podia is attracted to the receptor site. And here's the podia. This is considered the presynaptic neuron, the presynaptic coming before the synapse. And this tissue here, which is the dendrite, is referred to as the postsynaptic membrane. It's receiving it, and this is the neurotransmitter. Again, acetylcholinesterase can be secreted that stops the impulse from the dendrite or from the muscle itself. Okay, please call me tonight. Let me know if you've understood everything. Okay, I'm going to mention, I did talk about Schwann cells, talk about a little bit more about the major neurotransmitters, and then we'll talk about the oligodendrocytes. The major neurotransmitters, if you look on page 8, of course, we talked about acetylcholine, that's number 3, and acetylcholinesterase was the enzyme that stops the transmission. But 2 others we mentioned, and I want to mention these again to you, is epinephrine. The common name is adrenaline, which is released. And adrenaline and epinephrine is the hospital name. Adrenaline is what we commonly refer to it as. It's kind of a layman's term for that neurotransmitter. And epinephrine is the hospitals. It puts you in the fight or flight response. Another one is norepinephrine and noradrenaline. Now, they're both the same thing, just different terms, different synonyms. Norepinephrine and noradrenaline also work with epinephrine and adrenaline in putting you in the fight or flight response, except norepinephrine and noradrenaline works primarily on blood pressure, causing the smooth muscle walls of your blood vessels to constrict. And remember, when you constrict a blood vessel, when you tighten it up, you speed the transfer of the fluid inside, and you press within, and it speeds up the fluid. Hydrostatic pressure. We've talked about that before. So that's what they're aimed at. Okay. Next, I want to talk to you about oligodendrocytes. And big name, but they're really kind of cool. Now, they look different than Schwann cells. Oligodendrocytes do wrap around the axons of neurons in the brain and the spinal cord, but they do it differently. Now, let me show you an artist's rendition. Okay. This looks weird until you orientate yourself. And these pictures are beautiful in your textbook. So please, you paid for those beautiful textbooks and all those artistic illustrations and micrograph photos. So please make use of them. All right. Now, we've colored in blue for drama. Okay. Here is the cell body of the oligodendrocyte. Now, the oligodendrocytes are neuroglia found in the brain and cord, not in the PNS. But here's the oligodendrocyte. It has a nice large nucleus. This is the cell body. Here would be the cytoplasm that we call axoplasm, and it contains all the organelles that, I don't have to mention, you know the organelles. Now, instead of the neuroglia, the entire thing wrapped around the axon, like we talked about with the Schwann cells, there are arms, axoplasmic or cytoplasmic extensions that are coming off that cell body. In this illustration, there's 1, 2, 3, and 4. Now, here is the axon of a neuron in the brain. The artist has illustrated it in yellow, and here is the axon. Okay. And the extension of the oligodendrocyte, this arm or extension, is wrapping around the axon, just like the Schwann cells wrapped around the axons of motor neurons in the peripheral nervous system. It's wrapped around. So this arm is taking care of this neuron by wrapping around this part of it. This arm is wrapping around another axon of a different neuron. This one is wrapping around another, and this one another. So you can have an oligodendrocyte, which is a neuroglia in the CNS that has these axoplasmic, cytoplasmic extensions, which wrap around the axons of other other neurons. And what do? They keep the impulse in because the impulse can pass faster. They insulate, support, feed, protect. They're wonderful cells. Now, one thing that's interesting about neuroglia is for every neuron in your CNS and PNS, for every neuron, you'll have 10 neuroglia. So neuroglia there's many more neuroglia in the brain and in the spinal cord. The brain and spinal cord is full of fat, and that fat is in the neuroglia, and the neuroglia keep all this together, support it, protect it. They're wonderful, wonderful little cells, oligodendrocytes. Now, I don't want to not mention or show you another artist's rendition, and please look at these in your textbook. This one's colored differently, but it is also pretty cool. All right. Here's the oligodendrocyte, so it's labeled. Here's the nucleus, the axoplasm. It has 1, 2, 3, 4 arms. This arm is wrapped around the axon. Okay, the axon of this neuron. This one is wrapped around this neuron. This one is wrapped around this one, and this one a fourth one. So here you have one neuroglia, which is servicing 1, 2, 3, 4 neurons. 4 neurons. Neuroglia are important. Now, I'll show you a third picture, and then I'll mention something else, and then we'll review a little bit. Okay. Isn't this fun? Aren't you having a good time? I love neuroglia. Now, where is it again? Oh, here's the oligodendrocyte. Just look at the yellow one. Here's the nucleus, oligodendrocyte. The artist just showing you with an oligodendrocyte having 2 arms wrapped around this axon, and this one is wrapped around this one. Here's another one with 3 extensions, and this one is wrapped around this portion of the motor neuron, and this one by the lower portion of the motor neuron. Those are oligodendrocytes. Here is your neuron. There is the nucleus. These are dendrites. Here's the axon. Now, I did mention to you that oligodendrocytes are in the CNS and Schwann cells in the PNS, but you have altogether 4 different types of neuroglia in the CNS, and we'll talk about those some more. This one we'll talk about a little later. That's a microglia. These are ependymal cells, very important. These are neuroglia called astrocytes that give us the blood-brain barrier that protects our brain, and of course then the oligodendrocytes. So you have 4 different types of neuroglia in the CNS and 2 types of neuroglia in the PNS. Okay. All right. Let me see. That takes us to pretty much the end of lecture 2, and we did it in 54 minutes. What I talked about today, I gave you a little bit of a review from what we did the other day. I hope you understood it. I didn't get any phone calls, so maybe you did. And then what we're talking about today. I introduced you today to neuroglia. There are 6 different neuroglia altogether. There are 2 in the PNS, Schwann cells, and I didn't mention astrocytes, but I will mention that later. And then 4 in the CNS, and I talked about the oligodendrocyte in the CNS. Now, we do know that in the PNS, the Schwann cells are important because they wrap around and protect the motor neurons coming off the spinal cord, going to your muscles and glands. And these Schwann cells are wrapped around with a secretion, a heavy secretion of a lipoprotein known as myelin, MYELIN. And around that myelin is a sheath known as the neurolema. And the presence of that neurolema enables a good amount of regeneration to occur right there in the PNS. I wish we had a neurolema wrapped around the 4 neuroglia in the CNS. And so if we have brain damage or spinal cord damage, it can regenerate. But we don't. We just have that neurolema around the Schwann cells. I also talked about the nerve impulse. We said the nerve impulse is carried on because of 2 important electrolytes that we can't live without. Sodium, which is a positively charged electrolyte that likes to stay on the outside of the axons, and potassium that likes to stay on the inside of that neuron. And it's the exchange, sodium pumping into the inside of the neuron and potassium pumped outwards, that make that impulse occur, that cause the electrical impulse, or we call it also the action potential, or the wave of electrical negativity. I know I repeat a lot, but I do that because repetition works. And I want you to understand this and relate to these terms, know what these terms mean. But the interesting thing is that when a neuron is not being stimulated on the outside in the resting neuron, the resting potential, it has the sodium, positively charged sodium on the outside with the negatively charged potassium on the in. Now, the sodium and the potassium are the only 2 electrolytes that change position. The other electrolytes on the outside of a motor neuron and on the inside stay. So when many sodium are pumped in and just a few of the potassium, which have a positive charge, are pumped out, the polarity changes because of the presence of these sulfates and phosphates, which are electrolytes and have sometimes 2 electrons, causing that negatively charged portion on the outside of the neuron or on the inside of the neuron. Please read these in your textbook. Read this information about the neurons, the motor neurons, the electrical impulse, the electrolytes, the neuroglia, especially the Schwann cells, and what they do. And neurotransmitters, acetylcholine, acetylcholinesterase, adrenaline, noradrenaline, epinephrine, and norepinephrine. Read about those. And there's a lot more when we talk about transmitters, and I'll talk about some more of those, especially when we get into the brain. Dopamine is a big one. But I just want to introduce you to these 4 right now. Let me know. You can call me. I'm home. I can guarantee you that. So give me a ring, 761-3636, if you have any questions. So I'm happy to answer them, and I want to know if you understand. So don't hesitate. And we'll talk later. Thank you. That's been 59 minutes, almost an hour. Thank you for staying with me. And hopefully we'll talk later.